So with our next lecture, or the next lectures coming up, we're going to be doing endocrine and then starting on the nervous system. And these are our main communication systems throughout the body. We get coordination. We have these 36 trillion cells. We have to get some kind of coordination, and we got to get some control and communication across these distances. And so some of the main players in this is going to be the endocrine and nervous system. We'll also be signaling neighboring cells need to have a way to communicate with each other. So that's basically what this lecture is going to be looking at. Cell signaling or cell-to-cell communication. How do the cells communicate with each other to make sure everything's running properly? The two main ways in which we signal are electrical and chemical signals. Electrical is we're changing the membrane potential. We can signal this way through gap junctions, which we'll look at. But the main way in which we're going to be talking about signaling and we're going to be focusing on in this lecture is going to be the chemical signaling. Okay, this is where one cell sends out a messenger, chemical messenger signal, and the target cell can respond to it. Okay, and so target cells, to be able to, basically we have this communication. We got one, with our chemical signaling, we have one cell talking, sending out the chemical messenger, all right, and the other cell, the target cell, in which we're going to get a response in, it's got to have ears to listen to what the cell is saying, all right, and those ears are the receptor. Okay, so to be able to respond to the chemical messenger, a target cell has to have a receptor for that chemical messenger. Now this chemical signaling, it can happen locally, just within the same tissue, or it can happen over long distances. We'll talk about three ways with local communication. This is over short distances, our gap junctions using our electrical signals, our contact dependent signaling, all right, and then the major one, most prevalent, chemical messengers basically being released in the extracellular fluid, and then the target cell has a receptor to respond to it. Okay, these are three local communications, but again, this is just over short distances. So the chemical messenger gets sent into the extracellular fluid, diffuses out to the neighboring cells, but it's not going to diffuse all the way through the body in the extracellular fluid. Right. As I alluded to, the endocrine and nervous system, they are going to be involved in our long distance communication. Right. Endocrine releases those hormones, and those hormones, as long as a target tissue has a receptor, can respond to those hormones. And then the nervous system, although its chemical is released just in this small gap between its target tissue, the actual neuron, the main body of the neuron, can be several feet away. So we can get this long distance signaling. It's actually a combination of electrical signal followed by a chemical signal and how the nervous system is operating. Right, so we're going to go through all these. We'll go through the chemical messaging system, and then we're also going to look at the receptor and responses taking place when those receptors are being activated. So our first of our local communication is our gap junction, also known as electrical synapses. We're basically making these channels or tunnels between the cells. So the cells have to be right next to each other because they have to be connected with these red things here, these connexin proteins that make up these gap junctions. All right. This allows for small molecules such as ions, amino acids, ATP molecules to move across, but like large proteins can't move across here, so small molecules have access across, and so this cell essentially has access to the cytoplasm of the cell. They are basically connected. Okay. This is through the body. The neurons have them. The main place we're going to be talking on gap junctions is we'll be looking at this at those intercalated discs in the heart cells, allowing us to get, basically we get flow of ion here. The flow of ion can continue into the neighboring cell, so we can get this electrical signal across the cells. Okay, so we won't discuss much on gap junctions here because we're going to be mainly focusing on the chemical signaling. Okay, but they do exist. They exist in the nervous system. And one place we're going to, again, talk about them in more detail is the cardiac, when we get to cardiac and looking at how the heart functions. Another type of signaling, local signaling, juxtacrine or contact dependent signaling. This is where we have these CAMs, these cell adhesion molecules, which act as receptors. Okay, and they can be linked inside to the cytoskeleton, or they can activate intracellular enzymes. Okay, but they're on the surface, and what's binding is a molecule on the surface of another cell. We're going to see immune system, our B and T cells. Basically, they come in contact with molecules at the surface of, say, virus-infected cells. We'll present molecules out here, and this would say it's your T cell. There is receptors for these molecules that are presented at the surface here that activates, and we get signaling in the immune cell to activate the immune cell. Okay, and growth and interactions with the extracellular matrix. This is all ways in which contact dependent signaling takes place. And then finally, the last of the local signaling is our paracrine and autocrine, and this is where the chemical messenger is sent out into the extracellular fluid. Right. So we can talk to neighboring cells, and they don't have to be right next to each other, but they have to be in close proximity, because basically this molecule is just going to diffuse out. Right? It's not going to diffuse out all the way through the body, it's just going to diffuse out to the neighboring tissues. Right? And the two types, we got the autocrine and paracrine. Autocrines here, where you see the molecule is secreted out and it acts on a receptor on the same cell that secreted the molecule. The other is paracrine. Paracrine is the secreted molecule, chemical messenger, acts on a receptor of a neighboring cell. Again, this is local signaling though. It's not happening over these long distances. It's happening locally in, say, just an individual tissue. So the endocrine system, it's going to be one of the main long distance signaling systems. Chemical messengers here, are those hormones. And where do they get released? They get released in the blood. So our endocrine cell releases the messenger in the blood. It can travel via the bloodstream, and it's going to travel, basically it can travel to any cell. But what cells are going to respond is the target cells, and the target cells, what must they have? They must have that receptor. Okay. Cells without the receptor are not going to respond to it, but those with the receptor will respond, and we'll get a response in the cell. Compared to the nervous system, the hormonal system typically is slower since we're going out into the bloodstream. Okay, the neuron's going to release in this little synapse, and we get this rapid signaling, but the endocrine's kind of slow. We got to travel through the bloodstream, get circulated out to wherever tissue we're going to, and then we get a response. Difference between the endocrine and nervous system in duration is usually the endocrine system. The response is a longer duration than that of the nervous system, which is boom, signal, take away, boom, signal. So we get this rapid signaling via the nervous system. So the other system involved in that long distance signaling, our nervous system. Okay. Unlike the hormone system or the endocrine system, we're going to get this rapid, usually rapid signaling between the neuron and whatever target cells we're talking to, but usually the duration of the signal, it's quick to respond, but also quick in duration. It's not going to have this long-term effect on the cell compared to like the hormone or the endocrine system where the hormones have this longer effect. All right. How long distance? You see, even though the neuron, the distance between the neuron and the target cell where the communication takes place is just in this little small gap of extracellular fluid called the synapse. There's a small space, much smaller than what we can actually see with the naked eye, but this small space. But the key is that neuron cell body, this could be all the way up in the spinal cord, the cell body, and this signaling can be taking place, say, at the muscle at the big toe. Right, so this part of the neuron is basically be about several feet long. So we get this long control over these long distances. And different than the nervous system, there's just not a chemical signal being taken place. There is an electrical signal that gets converted to a chemical signal when we reach the target tissue or reach the synapse to communicate chemically. Right. Neurotransmitters, it's one of the molecules. Okay, it's the ones that are released in the small space in that synapse across from the target cell. Some neurons, though, release chemicals in the blood, so they act very similar to an endocrine in that they release their chemical messenger out into the blood. So that would respond similar to what a hormone would. It has to travel to the blood till it gets to the target tissue that has the correct receptor to respond to it. Usually slower in duration, okay, but the signal is usually, like a hormone, is usually longer in duration. So we looked at how the chemicals get released, and then right with the hormones get released in the blood, neurotransmitter across the synapse, paracrine and autocrine, it's into that extracellular fluid, and neurohormones, similar to hormones, as they get released in the blood. So that's the chemical, that's the one end of the signal, or the communication, in that we're getting the chemical out from the cell. And now let's look at what takes place when we actually get to the receptor and we activate. So binding of these chemical messengers to the receptor are going to activate those receptors. Okay, most signaling, not all, but most signaling going to take place through a pathway. Okay, we're going to activate a receptor, that's going to activate other molecules, and we get activation of other proteins, and we get a response. But just looking at here's our signaling molecule that binds to the receptor protein. Binding to that receptor protein is going to activate the receptor protein. And many times, activation of that receptor protein is going to activate some signaling molecules on the intracellular side. So what we're doing is we're taking a extracellular signal and we're getting a response or an intracellular signal taking place. So we're doing what we call transduction. We're transducing. Going across from the extracellular to a response intracellularly. Okay, many times activation of these proteins can activate or alter some key target proteins inside the cell, and ultimately we get a response. So we get from a extracellular signal outside to a response inside the target cell. Okay, this is just the general way in which this runs. Okay, many times the signaling takes place in a cascade, and we'll take a look at that for several of these. Okay, but this is how we get signal from outside the cell and get a response inside the cell. So knowing what the properties are of the chemical messenger, that can basically give us an idea of where the location of the receptors would be for this chemical messenger. We can have receptors that are intracellular receptors, so they can be inside the cytosol or inside the nucleus. But to be able to activate this means that the signaling molecule, chemical messenger, must be able to make its way through the plasma membrane. And what properties allows us to do that? Or small lipophilic molecules or hydrophobic ones that are more nonpolar. Okay. Steroids, your thyroid hormones, these are small nonpolar molecules that are able to zip through the plasma membrane, and that allows it to activate receptors that are inside the cell. Okay, the signaling through this is a little bit different than what we're going to see when we look at the receptors that are on the membrane here. Okay. And that these intracellular receptors, when bound, they act as transcription factor. And we know that word transcription, transcription is that first process involved in protein synthesis. So bound receptors are going to activate or cause gene expression of certain proteins. So the ultimate response of these activation of these receptors results in the production of proteins or synthesis of proteins. So this is a much slower response in that we have to go through transcription, translation, and so we won't get this nice rapid signaling. It'll be a slow response, but at the end we have proteins, and this will have usually have a longer duration of action than what we're going to see taking place at the plasma membrane. Okay. Even though these signaling molecules are lipophilic, they can also have receptors that are on the membrane, just like our lipophobic messengers will be responding to these membrane-bound receptors. Okay, and we're going to see there is usually a much rapider response when we signal through this mechanism. Okay, but lipophilic, they can be inside or on the membrane. Let's look at our hydrophilic or lipophobic molecules. These are molecules that are polar, usually in characteristics, and they can't make their way through the plasma membrane. They get turned away. So where must these receptors be? They must be on the membrane. They must have a binding site that is outside of the cell. Okay. And we have basically 4 classes of these receptors. We're going to have ligand-bound channel receptors. Basically, when the ligand binds, we're going to get opening of an ion channel. The most common or most utilized is G protein coupled receptors. So binding sites outside. This protein spans the membrane, and it is coupled with these G proteins that get activated when binding takes place. Right. Enzymatic receptors, and that binding activates an intracellular enzyme. And then integrin receptors. Okay. Binding can alter enzymes inside, or they are usually attached to cytoskeleton, and we can get some change in structure of the cytoskeleton inside the cell. Okay, so we'll look at these signaling, and we'll take a look at a lot of signaling, couple pathways, signaling pathways in which G proteins signal through. Okay, but again, hydrophilic or lipophobic, these messengers can't make it through, so they must bind to a site outside of the cell. But you can see what we're getting is an outside signal, and we're getting a response inside the cell. Okay, the quickest, very rapid acting of these receptors is ligand-gated ion channels. Okay, when the messenger, the chemical messenger, is not bound, the channel is closed. Okay. When the messenger binds, now the channel opens, and we can allow whatever ions through. Okay, some channels allow multiple ions, some just only allow one specific ion, just depends on the receptor. Okay, there are different ligand-gated ion channel receptors, and they will allow different ions through. The ones, if they allow sodium, potassium, or calcium ions, okay, what's the effect going to be is that it's going to change electrical properties of the cell. It's going to change what we call the membrane potential. Okay, usually the cell is negative 70 millivolts inside compared to outside, is more negative inside than it is outside. By opening some channels for these ions, it can change this value, make it more negative or make it more positive depending on which ion you're allowing to move. Okay, but this is very rapid because as soon as you open it, the ions are moving through, they're diffusing through and changing the membrane potential. So very rapid effect. There are also ligand-gated ion channels for calcium ions. Okay. Calcium signal we can have similar to what we saw with the other ions, is we can have an electrical signal because calcium coming in will make it more positive inside. But calcium can also, there are ways in which it can signal differently in the cell, and that it can activate signaling pathways called a calmodulin pathway that can alter protein activity in the cell. Okay. Calcium influx can also initiate exocytosis. Also, calcium can cause movement. We'll see it'll cause muscle contraction. And also calcium can cause release of intracellular calcium stores. Okay, so we can have a multiple effect with the calcium ion. It's not just limited to strictly changing the membrane potential like we see with the sodium, potassium, or chloride. So this is a key signaling molecule in that we can get multiple pathways activated via calcium coming in. But again, same is we have a chemical bound, and that opens the channel, and that will allow calcium in. You can see which way calcium's going, so we know calcium is high outside and low inside because it is diffusing into the cell. So another class of the membrane-bound receptors is those enzyme-linked receptors. And what that's going to do is when we bind the chemical messenger on the outside of the cell, it's going to activate an enzyme intracellularly. One of the more common of these receptors is tyrosine kinase receptor, and that it activates intracellularly a tyrosine kinase enzyme on the intracellular side of the receptor. So you see with no chemical messenger, the kinase is inactive. Binding activates the kinase, and remember from our enzyme lectures, a kinase, what does a kinase do? It adds a phosphate group to a protein. In this case, it's going to add it to a tyrosine amino acid in a certain protein. Okay, so you see the protein with its tyrosine residue, not phosphorylated. Once the enzyme gets activated, now we add that phosphate group to the protein, and that can activate the protein. That protein can go on to activate other proteins, or it can cause a response in the cell, but we get the signaling pathway cascade in which we get a response in the cell. Okay, so we activated, we have an outside signal, and now we're getting a response inside the cell due to the activation from this chemical signal. All right, but again, it is activating an enzyme on the intracellular side. Okay, another class of receptor, these integrin receptors. You see the receptor tethered to a protein that's anchored to the cytoskeleton. So binding of the chemical messenger here can cause activation of intracellular enzyme, but since we are tethered here, it can also alter the structure of the cytoskeleton. So the most widely distributed receptors are our G protein coupled receptors. Okay, and simply, what is a G protein? G protein coupled receptor is binding to the messenger. This would be a G protein. You can see it is coupled with these G proteins down here, and binding of this chemical messenger will activate these G proteins that can go on and either activate more proteins. In this case, this G protein is opening an ion channel. Okay, you can see binding of the messenger activates the G protein, and that activated G protein will go and open up an ion channel. Okay. Why do this instead of just having it as an ion channel? Remember, our ligand-gated ion channels do the same thing. They open the channels. Usually this is slower in pace, but usually there is a more sustained opening of the channel. Or the ligand-gated ion channel is very open rapidly and then closes. Okay, where this may be a more sustained opening the channel and allowing more movement of ions. Okay, this is one way a G protein coupled receptor can alter activity in the cell. Okay, opening the channel, depending on what ion, can change electrical activity, change that membrane potential we talked about. Okay. Or we're going to see G proteins can also signal through signaling cascades in the cell and can activate second messenger systems. Okay, one of those secondary messenger systems is our cyclic AMP messenger system. Okay, in this case, the G protein coupled receptor binding of the messenger here activates the G protein. Instead of going and opening an ion channel, the activated G protein goes and activates and what we call adenylate cyclase enzyme. Okay. That activates this enzyme, and now the adenylate cyclase catalyzes the reaction of taking ATP and turning it into cyclic AMP, which is called a second messenger. First messenger is this out here, and now we're converting to a second chemical messenger. Okay, in this cascade, you can see the signal transduction pathways form these cascades in which activation of one protein activates another, activates another, activates another, and we finally get a response or product. Okay, in this case, cyclic AMP in the cascade is going to activate a protein kinase A. Okay, as we talked with enzymes, is phosphorylation of proteins. Okay, kinase phosphorylates protein. It's going to phosphorylate this protein, and that can activate this protein, and we can get a response in the cell. Okay, you say why use these second messenger systems? What we see by using these second messenger systems, we're actually able to amplify the signal. Okay, it's going to make the signal much stronger in the cell. We can have one messenger and we're going to be able to activate, say, millions of proteins in the cell. So we get this from one messenger to millions of proteins activated. We get this amplification of the signal and we get a strong response in the cell. So that's one of the reasons of using a second messenger system, such as cyclic AMP here. All right, so let's look at this amplification. All right, here is that one chemical messenger. For simplicity's sake, you see the one G protein being activated, but there can be several coupled G proteins that get activated. Let's say in this case there were 10 G proteins activated by the binding of this one chemical messenger. Okay. If that 10 of those G proteins activate 10 adenylate cyclase molecules, all right, it is an enzyme, so as long as there's substrate there, it can catalyze reactions. In the cyclic AMP, say each of those, okay, each of these enzymes convert 500 ATP to cyclic AMP. That would get us up to about 5,000 cyclic AMP molecules. 5,000 cyclic AMP now activates 5,000 protein kinases. And again, our kinases, as long as there's substrate there, can catalyze reactions, and say they do 500 each. We can get us up to roughly 2.5 million phosphorylated proteins or activated proteins in the cell. So we went from one chemical messenger up to activated 2,500,000 proteins inside the cell. So we get this large signal, this strong signal in the cell. We're getting this amplification as we get through our secondary messenger system through the cyclic AMP. All right, this is one of the most commonly used secondary messenger system throughout the body. There are other ones. Besides cyclic AMP, there are other signaling pathways in which G protein coupled receptors signal through. One of these other signaling pathways signals through phospholipase C. Right, here is the G protein coupled receptor. There's the chemical messenger binding, and it's going to activate our G proteins. In this case, the G protein activates the phospholipase C. Okay. Phospholipase is going to break down phospholipids in the membrane and break them down to a molecule called IP3 and diacylglycerol, DAG. Okay. IP3 can signal to release calcium stores from the endoplasmic reticulum, and that calcium can result in cellular response. The DAG, the diacylglycerol, can activate phosphokinase C, which can then phosphorylate proteins, and again, phosphorylated proteins can go through a signaling cascade and again give a cellular response. Okay, I kind of wanted you guys to see big picture. The cellular response, it depends on what cell we're talking about. It can be very varied, but the signaling pathway is very similar in that we're activating through IP3 or diacylglycerol. Okay, but big picture too is we get a chemical messenger from the outside, the extracellular, and we're getting a response intracellularly in the cell. So we're getting the signal transduction through these receptors and these signaling pathways. Some properties of these receptors. When we saw the enzymes, you saw those enzymes. These are proteins binding to ligands, just as similar to enzymes binding to reactants or substrates to catalyze a reaction. They exhibit similar properties in that there can be saturation of the receptors. If all the receptors are bound, then the signaling molecules that are not bound cannot bind. So we can't get a signal from that chemical messenger. Just as if all the enzymes are bound, we can't get catalyzation of the substrates into product because the enzymes are all bound with saturation. Specificity, that is that these receptors bind to only certain chemical messengers. Some bind to one type of messenger. There's adrenergic receptors that bind to both epinephrine and norepinephrine. Okay, so they bind to a couple molecules, but they're not going to be binding to all chemical messengers. They are specific for usually one or maybe a couple chemical messengers. They can also exhibit competition, just as epinephrine and norepinephrine. They can compete with the binding to an adrenergic receptor depending on what how much concentration is there and so forth. If one is bound to the receptor, the other one cannot bind. So in a sense, it is competing for that binding site on the receptor. So the receptors we've been talking about, they have their primary ligand, primary molecule, chemical messenger that's going to come activate them. There may be other molecules that come in and activate, and if they give the same result, they activate the receptor and we get a similar result in the cell, they are termed agonists because they're eliciting the same effect as the primary ligand or a similar effect. Whereas an antagonist bind to the site, but when the antagonist is in play, there is no way for the primary ligand to bind, and what happens? It blocks the response from happening. Even if the ligand's present, it's being blocked, competed for by the antagonist, and you won't get a response. So many disorders or many diseases, this has kind of been a target for drug companies in that we can block or we can increase the activity of different receptor, different cell signaling. So many of your pharmaceutical company, the drug targets are agonists or antagonists for various receptors. I'm a neuro guy, so I'll use a neuro example here in that people with psychiatric disorders, they have delusions, hallucinations. It's believed to be a overactivity of the dopamine system in the brain. Different parts of the brain that are involved with dopamine signaling, dopamine binding to its receptor, this overactivity. So one of the drugs they use to treat these delusion and hallucinations is a dopamine antagonist. If you put that in, it's going to compete for the dopamine receptor, and if it's there, it's not going to get the response, so you won't have that overactivity, and that diminishes the delusions and hallucinations. On the other end, recreational drugs many times affect signaling. Dopamine is also part of your reward pathway, and a drug like cocaine is an agonist for dopamine. Okay, and your reward pathways, they have dopamine signaling in your nucleus accumbens, and so if you do cocaine, you're going to be active, but also you're stimulating those reward centers. So that's why cocaine is a very addictive substance, because you're firing that signaling in those reward centers. Okay, if somebody that takes cocaine takes a large amount, sometimes they have hallucinations, because what do we say? The overactivity of the dopamine system right? We use our antagonists to block the dopamine system, so sometimes if you take too much of cocaine, some of those people get delusions, hallucinations, and seeing things, probably because they are overactivating the dopamine system, which we try to squash with our antipsychotic drugs. All right, so moving forward, one thing I want to make note to is many of these chemical messengers. We're going to talk about acetylcholine. We're already discussing dopamine, some of these neurotransmitters. Okay, they are the same chemical messenger. What we're going to see is cells can have different receptor subtypes. And this is M for muscarinic. We're going to be looking at this when we get to the autonomic nervous system, in that acetylcholine can bind muscarinic 1, 3, 5, 2, and 4. It can bind all of these receptors, right? But what you see is you get differing effects depending on what receptor you are binding. Okay, so we can have one chemical messengers, but we can have multiple receptors, and depending on what receptor that cell has, depends on what response the cell is going to have. Okay. So keep that in mind, because when we get to autonomic, we're going to be discussing this, because our sympathetic and parasympathetic use the same neurotransmitter, but at different tissues, we're getting different responses. Okay, so we're not going to get the same response with each chemical messenger. Is not going to have the same response at every cell across the board. There can be differing effects depending on what receptor is getting activated. Okay. And finally, your cells can modulate receptor response. Okay, they can do what's called upregulation or downregulation. If there's an increase, if there's lots of chemical messengers, so what happens with, say, that dopamine, that cocaine with your dopamine, it's stimulating the receptor, so the cell's getting this strong, these cells that are targets for the dopamine are getting strong signal, right? And if it's very strong, it actually could kill the cell. So what do the cells do? How can they keep from getting such a strong signal? If you keep putting cocaine in, right, you're still getting this very overabundant signal, and the cells are being overstimulated. So what can they do? One of the things is they can downregulate. Downregulation, what do you see happening over time? Lots of receptors out on the membrane for, say, our dopamine to bind. We can reduce that. Okay, we can reduce the number, so then the signal is not as strong. Okay, that is why, hey, if you're taking cocaine constantly, okay, you've downregulated your receptors, and now you pull cocaine away. What happens usually with people on withdrawals? They feel depressed. Their reward systems aren't being stimulated, because what they do, they downregulate. Now just the natural dopamine's there, and they're not getting a strong enough signal. Okay. The other end, if the chemical messenger, right, is low, okay, there's very little signal. If the cell's not getting enough signal, it can upregulate, and you can increase the number. So even if the level of the chemical messenger is low, you will have more receptors to bind to get a stronger signal. So depending on what the levels are, the cells can up or down regulate, increase the number or decrease the number of receptors on the membrane, and that will affect the response inside the cell. It will affect the signaling transduction that is taking place inside the cell. All right, so that's it with cell signaling. We're going to move on, head on to endocrine. So that's a good point that we have this here, as we'll be using this signaling in our endocrine system.